You're listening to an Airwave Media Podcast.
Welcome to Unbiased Science,
where we bring scientific method to the madness.
We're your hosts, Dr. Jess Dyer
and Dr. Sarah Scheinman.
And this week, I am so excited to welcome a dear friend, brilliant scientist, science communicator, Dr. Yasmin Mohseni. And interestingly, we did a total pivot yesterday. We were supposed to talk about immunotherapies because she's this brilliant, badass immunologist. You'll hear all about her and her background in just a second. But then the Nobel Prize was announced, and it just happens to be in an area that is her expertise. So we're going to talk all about that in a sec. Before we dive in, just a super quick update in the public health world. So we're recording this, what is today? Tuesday, October 7th. Yesterday, all these headlines dropped because the CDC finally signed off on the ACIP recommendations regarding COVID vaccines. As a reminder, ACIP is the Advisory Committee on Immunization Practices. They had their 2-day meeting a couple of weeks ago. They talked about lots of things, including COVID vaccines. And unfortunately, this is getting interpreted by the media to mean that the federal government is no longer recommending COVID vaccines, but that is absolutely not the case. Mm-hmm. COVID vaccines are recommended for everyone 6 months and older. The difference now is that we're moving toward this shared clinical decision making. So it's an individual decision based on your conversation with your healthcare provider, but it does not change the fact that the vaccine is still recommended. You should be able to access a vaccine without a prescription wherever it is that you live in the United States. So we'll be doing more posts on this, but just wanted to clarify that things seem to get a little muddied in the headlines and a lot of people were confused. All right. Without further ado, Sarah, do you want to introduce our special guest?
I would love to. You guys, so today we are welcoming Dr. Yasmin Mohseni, who is a PhD and an immunologist with over 6 years of experience in the cell and gene therapy biotech space. She specializes in immunotherapy for cancer and immunoregulation. Dr. Mohseni earned her PhD in immunotherapy from King's College London, where she focused on using engineered regulatory T cells, or T-Regs, to promote immune tolerance in solid organ transplantation with applications to autoimmunity. She began her industry journey at Quell Therapeutics, advancing T-Reg-based therapies, and now works at A2 Biotherapeutics in the cancer immunotherapy space, developing therapies for solid tumors. She currently serves as the scientific lead within Quality, bridging analytical strategy, process and product knowledge, and improvements within CMC. Yasmin, welcome to the pod.
Hi, thank you for having me on.
Okay, just wait, because there are people listening right now who are like, what the heck did all of that mean? And I am right there with you. And I'm going to be totally transparent right now that literally a few minutes before we hit record, I thought that we were talking about T-Regs today. and Yas corrected me and she's like, it's T-Regs. And I feel kind of foolish right now. So she schooled me. Before we get into things, can we just take like a beat for a second? Because I am totally obsessed with Yaz's science communication page, and you can follow her at dr.yas, YAS _, right? Yeah. On Instagram. And we'll link to all this in our show notes. But one of the things I love most about your page is that you're so freaking smart, but you make science so approachable. You bring the human element. You tell us a little bit about you as a human being, as a person. I love it so much. And we're going to take one moment here to kfel. That is like Yiddish for I don't know, get excited about the fact that you just recently got married. Congratulations.
Thank you so much. I know, crazy. I feel like an adult now.
Right before this, she's like, I shooed my husband and my cat out of the room.
Yeah.
I'm like, I love it. Can we see the ring?
Yes, you can. You see?
Oh my gosh, it's so beautiful.
Yeah.
Congratulations.
He did good.
He did good. Bravo. He's a lucky fella.
I'm like, I don't want to see the ring. I want to see the cat.
Oh, they're outside. They're very clingy. So
that's what I'm excited about. I mean, good job getting married and all, but let's see the cat.
I'll have to get them in after. It might be a bit chaotic because
mine's around somewhere
with zoomies.
Oh, the zoomies. I love it.
We know about that.
So we kick off every episode with the same question. So what is something that's giving you, well, no, that's the closing question. Excuse me. Our kickoff question is, what is a news item that caught your attention in the health science space, and why should people care about it?
Well, very funny timing, because yesterday the Nobel Prize in Medicine or Physiology got announced, and it happened to be on T-Regs and T-Reg biologists. And I'm like, oh my gosh, yay. Like, I woke up to a flurry of DMs, text messages. I was like, people finally care about T-Regs and how powerful they are, and this validates it. And it just, I was absolutely over the moon, especially for these three scientists. Like, I've been tracking, I was always tracking Shimon Sakaguchi, and Fred Ramsdell actually was like a close collaborator of like Jeff Bluestone, who like we always like looked up to and also were kind of inspired by with the in terms of the immunotherapy space. And they actually have founded I guess I'm no longer in that space, but one of when I was at Quell in London, the rival or friend company, whatever you want to call it, friend Sonoma, and they're in San Francisco at the moment. So, and they're pretty much, advancing T-Reg based therapies. So it's amazing to see the, I've literally had I would say experts, but scientists who with a really renowned reputation tell me, well, we don't really think T-Regs do anything. And I tried not to like bark back at them, be like, you don't understand.
Well, this must be real validating for you then to just be like, suck it, folks. Actually, what I spent all of my PhD doing is really, really important.
Exactly. It's valid.
Hey, what are T-Regs? What are we talking about?
Or Tregs. It's so funny in my lab during my PhD, and like the lab down the corner of the building, we had like this massive inflatable T-Rex, and it's like like little giggle amongst the scientists. But
we have a great sense of humor.
Right, one way to remember how to pronounce it. So T-regs, or regulatory T cells, are basically cells in your immune system that stop inflammation. And that kind of sounds a bit counterintuitive when you think about immune system stopping inflammation. But if you think about it, you kind of need a system to switch things off as well, because you can't just have uncontrolled inflammation in your body when infection has been resolved. You need to switch off the immune system, otherwise you're at risk of hyperactivity, essentially like autoimmunity is a perfect example, or even the more aggressive cytokine storms, which we've sort of heard more about because of severe COVID-19 cases. That's where T-Regs come in. Their job is literally anti-inflammatory, to calm down and quell the immune response once everything has been cleared. So it's very, very orchestrated very well. They're extremely important because they have this effect, and without them, you would be literally riddled with autoimmunity. So they're very important.
Yeah, I mean, I think what a lot of people don't understand about the immune system, and I think you probably may have encountered this too, Yasmin, is that like whenever someone hears inflammation, they automatically think like, oh, bad, awful. But actually, inflammation means that your immune system is working and doing the job that it needs to do. It's kind of cleaning up all the garbage around your body and kind of making sure that the streets are clean. Like that's how I describe it to my students when I teach about the immune system. And it's important to do that, but you need kind of that system to be regulated a little bit, because it can't be kind of out of control inflammation. You want it to only be inflamed sometimes, right?
Yeah. And I think it's like definitely a misnomer when people are like, oh, how can I boost the immune system? It's like, well, you don't want to boost it. You want to get back to balance as quick as possible, you know? And like that's your body always wants to try and get back into that homeostasis, into that balance. So exactly. And that's where T-Regs are extremely important, because for those, especially with, I mean, I guess you could say chronic inflammation, but those that have autoimmune issues, or even like, if you think about like an overactive immune system, such as like fatal allergic reactions, you know, people don't remember how powerful the immune system is, you know, how powerful the immune system is. Like you know, it's, and not to get all morbid here, but it can kill you. It's lethal, you know, so we need these sort of regulatory systems in place to be able to calm down the immune system, to not have all this like off-target effects that are unintended in the body.
Yeah,
so,
I think the word balance you used is so key. Like that's basically why these regulatory T cells are doing what they're doing, is they're maintaining that balance and that homeostasis.
Yeah.
So what was the research that these Nobel Prize recipients are doing?
Yeah. So, I mean, when Sakaguchi, he was so, I nearly got emotional, and like, he was like so surprised. He was like, oh, you know, it's like, I think he said something about like, I expected it to be more for like, you know, those that are currently doing therapies or something of the sort. He's so humble. But essentially Sakaguchi was, I would classify him as the founding father of T-Regs. I mean, that could kind of be up for debate, but he's basically the person that identified this like unique cell type with this particular IL-2 receptor that, but it's basically specific like makeup, that if we didn't have them, it causes like a lot of autoimmunity, like a lot of autoimmune issues. And it maintains something that we call self-tolerance. So think of self like yourself, and tolerance. So like not rejecting you, not attacking you. So that's what Sakaguchi sort of uncovered in like the 90s. And then in the 2000s, Fred Ramsdell and Mary Bronco, and actually as well, Sakaguchi, further defined these cells as these T-Regs that have this main master regulator gene called FOXP3. So that FOXP3 gene essentially is basically what makes T-Regs T-Regs. And that's how they defined them as these specific cells. And I mean, that was kind of what paved the way for future T-Reg research. So
I'm so glad that they actually gave it to the founder of this stuff. And you mentioned that he discovered this for the first time, you guys, back in the 90s. I think it was like 1995 when like his seminal paper came out. Yeah. And where we're now like in 30 years later.
Yeah.
And now T-Regs are finally getting their moment in the sun. And I think it's exactly right that they should give acknowledgement, not just to people that are like using this stuff to create these fancy therapies, but actually to the person that first just made the discovery.
Yeah, I agree.
So I'm not an immunologist. A lot of what you're saying, and I know you're doing such a good job of taking something that is obviously super technical and making it more palatable for someone like me. But can you help me understand, like, what are the implications of this work, of this research? Like, what does this mean for people?
Yeah. So basically for me and personally, like my story, like how I ended up getting into immunology essentially was, I guess it's like easy to put it into that context. So just knowing that this is what T-Regs do, right? This is what like evolutionary, they were designed, and this is exactly what they're supposed to do. We can harness their power. We can harness their power when we have like hyper immune activation, we could call it like that, too much excessive inflammation. So, for example, in autoimmunity, something clearly goes wrong. Now, in a majority of cases, some experts might like, I don't know, maybe this is up for debate. I don't think it is. I think it's kind of universally accepted that in most autoimmune cases, a deficiency or like some sort of dysfunction with your T-Regs is kind of a major contributing factor. Even other regulatory type immune cells as well. There's something going on because they're unable to control this inappropriate immune response against you, i.e. that self-tolerance we spoke about is broken somehow. But then imagine everything is behaving appropriately. Everything's behaving as it's meant to and, in order. What is the job of the immune system? Its job is to basically reject anything that's foreign and attack and try and remove anything that's not self or non-self, like an organ transplantation. So if you get an organ transplant, a perfect litmus test that your immune system is working as it should is if it tries to reject the organ. Unfortunately, that defeats the point of the organ transplant, right? You want to calm the immune system down. So what actually what my lab and others identified before I joined was in liver transplantation, they found that in 1% of cases, maybe even less than that, there was this spontaneous acceptance of the liver transplant, that they're like, how, why? And we know the liver in general is kind of like an immunoregulatory organ, but what they found was that there were a bunch of T-Regs there. So are the T-Regs actually basically standing guard, and any cheeky immune cell comes to take a jab and a swing at the organ, it's like, nope, boop, switch you off. So that's kind of where, we started to go with, maybe we could use this as a type of therapy. On the flip side, Fred Ramsdell and Jeff Bluestone have obviously founded Sonoma, I think it's Sonoma Biotherapeutics. But anyway, Sonoma is based in Northern California. They were looking more of the context of autoimmunity, like type 1 diabetes, for example. It's a similar concept with, if you have obviously an organ in yourself, not a transplant, but an organ that's being attacked, can you drive T-Regs to go to that organ and protect the organ? Maybe for IBD, it's the gut. Maybe for MS, it's the nervous system, So that's kind of where we're thinking, wow, maybe we can use this as a type of therapy. So that's the standpoint that I came from and that I moved into for my PhD, which is actually my PhD. I'm an immunologist. I thought it would be cool to tick the immunotherapy box when I was getting my diploma. And I'm like, well, technically it's immunotherapy. I don't know if I regret that now, but it is, it's a type, using your immune system as to create a therapy. So that's the potential. And right now there are a bunch of T-Reg based therapies that are in clinical trials for these specific indications.
Yeah, I think what's so cool about this too is that you're harnessing things that your body is already producing in order to help itself. We all have regulatory T cells, and they serve a very important function to maintain that balance between attack and restraint that the immune system is so known for. And, that's I think probably why it got the recognition that it deserves is we're not reinventing the wheel here, folks. We have these cells in our bodies.
Exactly.
So okay, bear with me for a second. So you talked a little bit before about autoimmune issues. You talked just now about organ transplantation. What about the big C, cancer? Could there be applications to cancer?
So typically T-Regs are actually not good news in cancer. And that is, I did this little bite-sized post, but it just proves how complicated the immune system is and how trying to find ways to reset it or somehow manipulate it yourself really isn't easy. Because, if you try and quell the response, maybe you'll come back with more inflammation, as I found during my PhD, which was quite painful, but that's a story for another day. Go read my 500-page thesis. But in cancer, obviously, what do T-Regs do? They suppress. Now in cancer, actually, tumors manipulate T-Regs to protect them. So what T-Regs, especially solid tumors can do is they can create this fortress called the tumor microenvironment to basically protect themselves. And in that tumor microenvironment, which I like to think of it sort of a cartoon villain that has, the sort of sharks protecting the castle, on this hill that you just can't penetrate whatsoever. T-Regs are also there protecting the tumors. So they're the manipulated generals, I guess we could call them. So there are therapies that also look at depleting T-Regs or getting them out the way somehow in the case of cancer. Now, that doesn't, I know that for any like cancer patients that are listening, they're thinking, oh, maybe, oh my goodness, maybe like that's the strategy. And like, you know, that is being explored as well, just so we're clear. But then the downside is, of course, is if you get rid of T-Regs, then you're at risk of mass autoimmunity in the body. So it really is this fine tuning of the balance there, you know? So.
Wow. That is so fascinating. And is there a way, I guess this is really, please pardon my ignorance, but like, is there a way to come in and sort of specifically target T-Regs? Like, I don't know. I don't know how to explain what I'm saying. Like when I think of, I don't know, chemotherapies and stuff like that, it feels like, I know they take the bad, but then they also suppress some of the good, right? You know, that's my very lay interpretation of it. Is it like, are we trying with T-Regs to like go in and have like a very, like a surgical targeting of those specific cells?
Yeah, I mean, that's kind of where the targeted immunotherapy comes in, which we could talk about another time of part two, because even like chemotherapy, they're making that super specific targeted now. Even like using like antibodies, like to basically we like call it ADCs, so like antibody drug conjugates. So basically like you load the chemo onto this like antibody or engager, and it drives straight to like the tumors essentially, because you can look at specific markers that tumors might have, and that's how you target them. But that's like a conversation for another day. With regards to T-Regs, similarly, but as like, for example, anti-CD25, we call it antibodies. But one of the big downsides of T-Regs, now we can definitely fact check this, but I'm fairly certain that nothing has been evidently discovered yet on this, is defining them specifically by anything they have on their surface that makes them distinct from other cells. So we know that main gene I spoke about, which was one of the reasons why the 3 Nobel laureates won the Nobel Prize, was the FOXP3 gene. But that is, I mean, even that's not actually, ironically, unique to T-Regs, because other like T cells can like transiently have it switched on. But it really comes down to a specific region in it that if it's not methylated, then we know that it's typically associated with T-Regs. But that's inside. That's inside the cell. We need something like a little like arm or something sticking out on the surface to be like, hi, I'm a T-Reg, come to me. I
like
think
a little flag.
Right, a little flag, exactly. Like, hi, come to me, pick me. And I think, I mean, everyone is sort of researching using like real intense mass cytometry to be able to look at every single potential, you know, marker there could be. I think there is actually one patented at the moment, which it kind of sucks because it means none of us can go for it. But as of when I last checked being in the field, which I left 3 years ago, I haven't seen any updates in this, but we can check and like maybe add in the show notes. But that's what it is. It's like trying to find something to specifically harness the T-Regs or like specifically deplete the T-Regs. And as I say, the strategy at the moment that's typically being used is this anti-CD25 approach, because T-Regs have this CD25 flag, but the downside is also activated T cells, like the killer types, can also have this CD25 flag. So you don't really want to get rid of those either, because they're the ones that are also coming in to like drive out the cancer. So it's constantly keeping us puzzled and on our toes for sure.
So, sorry, Sarah, were you going to?
Oh, no, I was just going to say, I mean, it sounds like, I know that these regulatory T cells, they're very, very complicated, and they're really kind of smart, but I guess if I'm listening to this, I would be thinking to myself, like, how do these cells, like, quote unquote, know, like, when to suppress the immune response and, like, when not to, I guess?
Yeah. So, I mean, they're self-reactive, typically. That's what they say about, like, you know, T-Regs. There was this really cool paper that came out in Science, actually, recently, I think it was a few months ago, where they managed to identify that essentially when you have infection, actually T-Regs just automatically begin to amplify because they're ready to calm down the immune system. So even like if you have an immune response, not only are your killer immune cells and all the other immune cells that are in charge of of clearing the infection, amplifying, but T-Regs at the same time are ready, so they're in return amplifying. So it really is down to if there's an infection somewhere in the body that does drive the T-Regs into the area ready to, and they have many signals as well to be able to get them there, and they have many sort of sensors to be able to get them to drive into the area. But that's also part of what my research is how can we not use something called polyclonal T-Regs, which are basically T-Regs that they're not really specific. I mean, they're your T-Regs, and they, we know that they traffic or travel to a site of inflammation, but how can we ensure that they don't get distracted elsewhere, that they specifically go to where we want them to go? And that is through, focus. And that's through engineering strategies, which was one of the approaches I took during my PhD.
So what would you say is the buzz right now in this field? Like it sounds like there's a million different applications and things that lots of brilliant people like you were focusing on, but what's something that's really buzzy, where a lot of people are focused in this space?
Definitely autoimmunity. So that's the area that everyone is very, very interested in. Now, the irony is that there was buzz in using a slightly different application of the tech, CAR T cell therapy. So harnessing the power of the killing immune cells, which we use we use to basically in trials for cancer. I know it's FDA approved for like a bunch of the blood cancers. But the logic is that if you kill all the naughty cells, such as all the B cells, like for leukemia, then in theory, you get rid of the blood cancer, right? Similarly, if you kill all the B cells that are making antibodies against you in an autoimmune response, maybe you send a patient into remission for their autoimmune disease. And that's kind of what they've been seeing with this CD19 CAR T approach. This New England Journal of Medicine paper came out that kind of caused a massive buzz and made everyone interested in autoimmunity. And I find that interesting because I'm in that space now for cancer, but as a T-Reg biologist, a lot of us are thinking, we don't have to just kill everything, mate. Like we can, like, what if? And I mean, maybe it can be a one and done. If you clear all the autoreactive B cells, you clear the autoimmune response. I am not a believer of that as a T-Reg biologist, as a T-cell biologist. T cells are also naughty when it comes to the autoimmune response. They also are targeting, your organ when they shouldn't be. So what we're going to do with the T cells? What we're going to get rid of the T cells as well? Like you're just going to try and do a mass immune reset? I mean, you can, but that is going to be not just through following supplements or diets, hardcore chemotherapy and full body immunoablation. And even that is just not worth the risk and, only approved for certain things such as blood related disorders. But yeah, so that's kind of the buzz right now. I think the knock-on effect now, I am predicting of the T-Regs winning the Nobel Prize, or rather not the T-Regs themselves, but the pioneers winning the Nobel Prize, and those in the cell and gene therapy space aware of the buzz of autoimmunity. Hopefully everyone will put two and two together and kind of catch up and be like, wait, we can now use T-Regs. And there are big players in the field at the moment, including biotechs, that are doing this. So obviously Sonoma, Fred Ramsdell is at Sonoma. And also the company that I came from in London, which was founded by my professor and 5 others in Europe, Quell Therapeutics. And that is harnessing the power of T-Regs to stop autoimmunity. So that is the buzz, that's the application. And I mean, if you think about it, like with CARs, so they stand for chimeric antigen receptors, they're fully customizable. So you can swap in and out whatever you want. You can make it, in theory, target whatever you want. And once it gets there, induce whatever response you want. Just it's all to do with engineering. And that's why like this dawn of genetic engineering and cell therapy is just incredible. But obviously you have the intention, the theoretical of like, we can get this CAR T-Reg to target the myelin, for example, for MS. But 1, will it get there? And 2, if it does get there, is it powerful enough to be able to protect it? So, there's theory versus like in practice,
Sure.
Step aside, Ethan. I'm in love with Dr. Mohseni. You're so unbelievably brilliant. This is exciting stuff. This is honestly probably one of our most positive and uplifting episodes because it's
of the season for sure.
Of the season for sure.
Absolutely.
We're always like doom and gloom. Everything is terrible, and now it's like, look at this amazing scientific breakthrough that's opening up the door for these incredible treatments. And oh my gosh, so cool. Well, I guess that leads us, and maybe you've already answered this question, but maybe there's something unrelated to what we've talked about. We like to close every episode with, what is something in the public health, science, medical space that gives you hope right now?
So I think it can be applied to like either the T-Regs or the T-cell immunotherapy space in general. Like I classify myself as just being an immunotherapy in general now. It is that we are curing the incurable, I think. I think that's something that gives me massive hope and makes me extremely excited. There were terminal cancers initially that are now, if we're catching it early, are no longer terminal. Like if you catch it early, we can treat it. And that's what's getting me excited. And even for like autoimmunity, now there are new class of biologics and drugs that are coming out. And who knows with T-Regs as well. Like I'm obviously biased. That I think, this is unbiased, but I am biased, that that T-Regs are going to play a massive role and do play a massive role. So I'm really excited because of the power of engineering and how we can literally customize. It's crazy how we can customize now. Just create a receptor and then whack it into the immune cell. And then there's in vivo engineering, which is a whole other topic on LMPs that we can talk about another day. And, COVID vaccines have got a massive role to play in that whole research.
Oh my God.
I'm really excited to see how we, cure diseases in the next 20 years.
That's amazing. Yes, thank you so much for joining us. Can you remind folks where we can follow along all your incredible work?
Yeah, so I'm on Instagram. It says at Dr. like full spelling DOCTOR .YAS_. And that's on Instagram. I also like cross post a lot of the videos on YouTube. I keep toying with getting on Substack. I prefer making videos because it's just more fun and easier for me. But I'll keep you posted to do that.
Amazing. Well, thank you so much for everything that you're doing. It's really incredible stuff.
Thank you
for joining us.
Sarah,
all
you
right.
want to take us home?
I would love to. Thank you all for tuning in. We hope you learned a thing or two. I certainly did. Please join us next week on your trusted source for no nonsense, just science.